glad to be here, so thank you for signing up for this hour. And what I'm going to do in this hour is really just kind of tell you about my life as a scientist, what it's like, why I came to do what I'm doing, and sort of what the day-to-day is all about. So the title of the talk was Building Organs One Gene, One Cell at a Time, but the sort of broader scope is regenerative medicine. So what I hope to do is I didn't know about this job when I was in high school, and so you may not either, and to just give you a flavor of what it's like to be a scientist and how you get to do it, all right? So I'm showing you this beautiful image, very artsy at the very beginning, but what this is a section through a pancreas, and these are all cells of the pancreas that are doing different things. The red cells are blood vessels, and this is part of what we study. My interest is really trying to understand how do cells make a decision to have a certain function. Why did this cell here decide to be a blood vessel, and this cell here decide to be a cell that's going to make digestive enzymes in the pancreas? Why is that? Why do you all pretty much look exactly the same? To an alien that would come down, you look exactly the same. You have two eyeballs, you have one brain, you have one heart. How did that happen? Why are we all so similar? And why did it happen right every single time? Well, it's genetically encoded. And so trying to understand that genetic blueprint is really what I'm interested in. So if you want to pursue this road, I'm going to tell you how I did it and how I suggest doing it. One of the ways is take these pre-college science classes, immerse yourself. I actually wanted to be a history major, and I wanted to teach history. That was sort of the way I viewed my life. Going to Europe, going to all the old churches and visiting, that's what I really thought. But my dad said, no, he says, you have to take a full range of subjects. And so when I went to college, I took calculus, which I hated. I took organic chemistry, which I hated even more. But I happened to sit down in a class where the guy was teaching molecular biology, and that was all about DNA and how DNA makes decision to become expressed in the RNA and then into protein. And I realized, I said, that's history. That is understanding where we come from. And I called my dad and I said, can I change my major? And he was like, yes, of course you can. So that's what I decided. So I suggest if you're interested in science, and we need scientists, take those classes and immerse yourself. You don't have to make a decision right away. But there is a great need, obviously, for all kinds of scientists. This was an old slide that I got, I think it was in 2010, but this was just some trends, and for the back, maybe you can't read them so well, but these were the fastest growing jobs back then. Biomedical engineers, and I'm listing some of the salaries there to show you that you can make any decisions. You're going to be paid. Network system, data communication, home health aides, these are all in the range of science, right? They're STEM related jobs, and there are so many of them and a variety of them. Just along those lines, this was the projected percentile increases in STEM jobs for various fields: mathematics, computer systems, software, medical scientist, biomedical engineer. And I didn't know it, but really that's what I've become, as a biomedical engineer. And I never took in my life a biomedical engineering classes. I became it because I became interested in it, and that's where my job led me. So there's a great need. So what I'm going to do in the next couple of slides is just to show you three things that occurred to me last night when I was putting this talk together. Needs out there for scientists in the life sciences. One is feeding a growing population. We need scientists to figure out how we're going to do this. I always include a graph, and I forgot to do it, but what I want to tell you is that it took up all of human evolution, all of human history, to get to 1 billion people on this planet. It took up until about 1800. All right, so that's a long, long time, building up very slowly. And then in 1800, we started having steam engines, and the Industrial Revolution kind of got going. And then it took 100 years to get to 2 billion. And then we really started factory production, massive food production, industry. The American Industrial Revolution really hit. And then it took 50 years to hit 3 billion, and then 25 years to hit 4 billion, and then 12 years to hit 5 billion. And we're now almost at 8 billion. So if you looked at a graph, it's flat for most of human evolution, and then it exponentially goes up, and we are at the top right now. So the planet is limited, and we need to be able to feed everybody. So we need scientists to be able to figure that out. And with innovation and with science, we can do it. We can figure out the ways to feed the population. Another thing that we need out there, scientists for, obviously, is disease. And I love showing this image here because this is a cancer cell that is being attacked by what's called a natural killer cell, and we all have natural killer cells. And right now in your bodies, probably half of us have cancer cells, and we don't even know it because we're healthy and we have a great immune system, and those cancer cells are being gotten rid of for you without even your knowledge. And so, scientists have made great inroads. The number of, the percentage of cancer is actually going down. We're doing a really good job. Not all cancers are the same, obviously. There are different cancers of different kinds. But one of them that's really important is leukemias. And that is really well figured out at this point. And this is the same child undergoing chemotherapy, in which case they wipe out his entire blood system, his white blood cells, get rid of those cancerous leukemic cells, and re-inject him with new stem cells that repopulate his body with new non-cancerous cells. And that's the same kid just months later, and he's doing fine. So we need scientists for disease, obviously, in this growing population. And then just the third example that I thought was really timely was we need scientists for the threats to our biology. And so you guys may be following some of this coronavirus that's just emerged on the other side of the world that is a real concern. We really started noticing it in December, but we weren't told about it, and it exploded. But what I wanted to show you here is the virus itself, which is a non-living particle, which infects cells in the respiratory system and replicates, makes you sick, and is killing people. And so here are just some articles that are coming out, and I'm learning a ton about this on the news, on Twitter. White House asked scientists to investigate the origins of the coronavirus. And how are they doing this? To investigate the origins, they're looking at the DNA. That's how they're doing it. Down here, scientists explore coronavirus treatments. More need for scientists. So this is the need you would be fulfilling should you choose to go in that route. Here is why molecular biologists in particular are being useful with this coronavirus. This is just to show you how much we understand now that we didn't used to about viruses. This is a coronavirus. These are the particles on the outside that allow attachment to your cells for them to get in. Here's just a cutout showing you all the proteins on the inside, and then the DNA that's on the inside of these cells, or RNA. And here's a recent tweet, January 31st. The Institut Pasteur in France becomes the first in the world to sequence the complete genome, the complete gene sequences of this coronavirus. And this is it. This is the entire coronavirus DNA. And so it's been interesting for me to go on here, and all of this is publicly available data. And understanding, we're learning how portions have recombined between different types of viruses. So the coronavirus, which is a typical sort of cold virus or flu virus, is recombining with really bad things like SARS. And they're looking at these sequences and saying, weird, looks like some of them might come from bat. It looks like some of them, these sequences might come from pangolin. You guys know what pangolin is? It looks like an armadillo a bit. But why this has happened is that mutations have occurred, and mutations is a natural part of DNA. We're thankful for mutations. If there hadn't been mutations, we might still be teeny little organisms in the ocean. But mutations can be either beneficial or they can be deleterious. And what I'm showing you with this little video here is just the normal kind of mutations that happen during cell division. So early on, when you're born, at the very beginning, before you're born, when you're conceived, there's your dad's DNA and your mom's DNA, and there's lots of recombination and mutations that occur that mix it up. And that's why you don't look exactly like your dad or your mom, and things sort of mixed up. But what they're showing you here is a chromosome, a green one and a red one, and they've exchanged little bits of DNA. It's a natural behavior that DNA wants to do, is to exchange pieces. And that's good sometimes, and sometimes it might be bad. But so these were just 3 examples to show you the usefulness of scientists in society nowadays, and there's many more examples. But So now what I'm going to do is just kind of take you through my science and my road leading up to what I do now. So I actually I was born in California. My parents were graduate students at Stanford, and my mom was French and my dad was American. So they moved to Quebec, where both English and French were spoken, and which was a shorter plane ride to France. But then I decided I wanted to be educated in English, not in French, and so I moved to Texas. And my dad was a teacher at this time at the University of Texas, even though it didn't give me any break on tuition. But anyway, it was an amazing experience to learn English and to become part of UT Austin. It's a huge school. You can really get lost there. This is the classes. When you go to these big state universities, you can have classes up to 500 people, right? And so I sat in these classes, and at the time, like I said, I thought I wanted to be a history major, but my dad had forced me to sort of take a variety of classes. And like I said, I took a lot of classes I didn't have interest in and I wasn't very good at. But when I sat in this class that was called molecular biology, it was taught by a guy who was interested in why heart forms, how do cells make decisions to become heart. And he was teaching us all about the different genes that would turn on, how the mutations would happen. And I just went, oh my gosh, this is what I want to do. And so I called my dad in my third year of college and said, can I switch majors? And he said, of course, that's what this was all about. And so I ended up getting a BA in history and getting my Bachelor of Science in molecular biology. Making my way through college. And making decisions about what I wanted to study. And what I really decided I wanted to study was DNA and genes. I wanted to understand, in every single cell in your body, there's the same exact DNA. And my DNA and your DNA are really, really similar, 99.9% similar. And so why do we look slightly different from each other? That was one thing I thought was interesting. How did I come to be? And so I became interested in how this blueprint, which is in every single cell in my body, how did it direct some of my cells to become eyes, and some of my cells to become heart, and some of my cells to become blood vessels? And it seems like a huge question, but it's tractable. We can ask these questions. And DNA, of course, is inside of cells. And so this is an image here of a cell dividing. And so you can see it at the beginning. There's one nucleus. The nucleus is a sort of little pocket within the cell. So you guys have all had cell biology, or has anybody had molecular biology? Any of your classes taught you about genes? Yeah, a few. Okay, so you guys know what it is. But so when a cell divides, its nucleus has to break down. Its chromosomes, you can see in this nuclei here, the chromosomes are very diffuse. But when the cell divides, they get turned into chromosomes. They get sort of compacted. And then the cell it replicates, and then the cell divides, and these two exactly similar sets of chromosomes are identical, mostly identical, get pulled to each side. And the green here, does anybody know what the green is? What are those little hands? Huh? Spindles? Yeah. It's cytoskeleton, basically, right? And the cytoskeleton are just little filaments that organize things. It's like the skeleton of the cell. So how is this happening correctly every time? If it doesn't happen correctly, the cells on each side won't be viable if the chromosomes are not separated properly, right? And you can see it. It happens incredibly quickly. And then once the DNA gets brought onto each side, you can see the cell is actually constricting right here. These filaments, the cytoskeleton here is constricting, and the cell's going to pinch off, and the two cells are going to be, mostly identical. So this is an incredible process. And although we know a lot more than we used to 50 years ago, there's still so many questions that we don't understand. And so this is the place that I want to make a difference, in which I'm excited to study. So kind of recapping, this is a cell up here, and there's lots of stuff in the cell, different organelles, and this is the nucleus, and the DNA is in the nucleus, and the DNA can either be sort of diffuse, or it can be condensed into chromosomes, and depending on how condensed the DNA is, and one of the sort of ways that the DNA is condensed is using these histones or proteins that sort of wrap it up. One of the ways you can control which genes gets expressed is by how loose the DNA is. So loose parts of the DNA will generally be expressed and turned into RNA, and then tightly wound up parts of DNA means that they're not going to be expressed. And so to me, the interpretation of this blueprint is absolutely fascinating. So how do you package it up? How do you express it? And when people talk about gene expression, this is it right here. The pink is the DNA. The little blue ball over here is called a polymerase, and it's reading the DNA. And then the yellow thing here is the RNA that's being made from that DNA. Now, most of DNA is actually not read and made into protein. It's not called coding. Most DNA is actually a lot of regulatory regions that dictate the parts that are going to be expressed. So all of that to me is fascinating. How does that happen? And then once you make this RNA, the RNA is going to be then read itself, and then you're going to have protein made. And those proteins are really going to help the cell decide who it's going to be. Is it going to be an eye cell or a heart cell or a muscle cell? And you might say, well, that's really nice and cute, but why do we care about what the cells are deciding? Well, if you wanted to make an organ in a dish, which is one of the things we're interested in our lab, you need to understand how this is happening. How is the DNA being interpreted, and how are the cells making these decisions? And one of the representations of the work that I've been showing for a long time is really this. My work is encapsulated by this slide. It can be called developmental biology, or how do cells divide and become different from each other in the early embryo. So the process of sperm meeting egg, that's two cells, dad and mom, is the very beginning, right? And then how do you go from this fertilized egg, which is really, really tiny, it's not even a millimeter, it's 100 micrometers. What happens here that leads up to the creation of this amazing thing, a complex multicellular organism with lots of functions? It can move, it can think, it has thousands of different cell types. So how did that happen? Do you ever think about that, how this happens? It's an incredible process, and you can break it down and figure things out. So that's my job, is to figure out that question mark. So it can be, some of those steps can be represented here. Here is a single cell, and fertilization is happening. At this point, when there's mom and dad together, it becomes a zygote. And now the cell starts to divide. It goes from basically one cell, two nuclei, to two cells. And then it divides more and it goes to 4 cells. It's washed out here, but you get the idea. Then from 4 to 8, each cell divides into 2. So as this is happening, you can imagine these cells are all becoming different. And we know that some of these cells here will give rise to the front of the embryo, and some will give rise to the back of the embryo, and they're sorting themselves out. And then at least for humans and mammals, higher level here, they start to sort out in this way, and you make a blastula, and the cells on the inside are you, and the cells on the outside are actually going to become the placenta. And so then they reorganize, and this is the gastrula here, where all your tissues are really making decisions and forming. And that's you, the little red part here, and these are all of the extra embryonic membranes. And there they attach. These parts here are attaching into the uterus, and then here you are being sort of created. So this entire process interests me. How do those cells make different decisions? How is the DNA being interpreted differently in this cell versus that cell? And even within the single embryo here, this cell versus that cell. Because I want to be able to recreate it in a dish. And so I like to show this slide here because we have many tools as molecular biologists, as developmental biologists. And one tool that has been an amazing thing has been the discovery of green fluorescent protein. And this was isolated from organisms in the ocean, jellyfish, that make this green fluorescent protein to protect themselves, really, to scare people that are going to prey, not people, but organisms that want to eat it. And in fact, this summer, my family and I went to Mount Desert Island Biological Laboratories to do some research for half a semester. Anyway, we were leaning over the pier and we were touching the water. And when we did that, you could see all this blue phosphorescence. Has anybody ever been to Puerto Rico or been swimming at night in the ocean and seen this? It's an amazing thing. Well, what those organisms in the water are doing is that they're flashing some fluorescent proteins to try and scare. They think I'm trying to eat it. I'm disturbing it. But we have cloned this gene, and now we can put it into organisms. And so my kids love these images here. This is a bunny rabbit that has green fluorescent protein in every cell. These are mice here. They're all siblings, and some of them have the gene and some of them don't. And so this was engineered. The protein was inserted and engineered, and you can either put it in every cell or you can put it in specific cells. This is a worm here who has it only in the neurons. And so this allows us to see specific cell types and separate them from other cell types. So in my lab, we have many different versions of this where we have it only either in the pancreatic cells or only in the blood vessel cells. So it's been a really amazing tool as a molecular biologist. But so all that just to say, we're doing some exciting science. You probably see it on the cover of many different magazines. I've shown you some of the tools. I like to show this image here because it's also kind of in my field. Does anybody know what this is? I mean, it's obviously an ear, but do you know why it's sitting in a dish? Huh? I heard it. Huh?
Did they grow it?
Yeah, somebody was growing it. Why do you think you would want to grow an ear like that in a dish? Yeah?
In case you want to have like surgery to like replace it?
Yeah, exactly. So this was done, in fact, for soldiers out in the field, right? So if you get parts ripped off from a bomb or something, you can replace them. And so how this was done was actually, we know a lot about cartilage and we know a lot about ECM or scaffolds, extracellular matrix proteins. And so somebody took these proteins and they molded into the appearance of an ear, to the shape of an ear. But then they knew about stem cells for cartilage, and they knew about stem cells for skin, and they knew about stem cells for blood vessels, and they seeded them on this ear. And now this ear can be attached and allowed to sort of integrate, and it sort of helps to replace an organ, right? So we would like to do this. This is a simple one, but we'd like to be able to do this with a lot of different tissues. Anybody have a family member that had a heart attack? Yeah, okay. Does anybody know what a heart attack is caused by?
Clottage.
Exactly. So your heart is one big pump, but it's muscle. And in the muscle is lots of blood vessels. And if that blood vessel gets clotted and gets occluded, then all the tissue that's supposed to be fed by that blood vessel dies. And that's what a heart attack is that the heart suddenly, a big portion of it dies and then it stops beating because it doesn't like that dead part, right? So we would like to be able to make new heart tissue to replace that dead tissue. Right now we do all kinds of bypass surgery and we put stents and we try to deal with it in different ways, but one part would be to actually regrow the heart. So that's in the field of work that I do, is to just make tissues in a dish to be able to replace them in patients that need it. So at this point, I'm just going to go back to sort of my history. I'm going to tell you about my Ph.D., I'm going to tell you about my postdoctoral work, and I'm going to tell you about my work right now at UT Southwestern. And you'll kind of see my interests as I sort of move through this. So these images represent my Ph.D., and this was Paul Krieg, who taught that class that I was just like, oh my gosh, I need to change my major. He taught about DNA and how it affected the cell's decision to become different things. And his lab was a lab that did work on heart, in fact. And I went to his lab, but I didn't want to work on heart, and I started working on blood vessels, and he said, sure, fine. And one of the things that I did was to clone a gene called VEGF receptor 2, which wasn't cloned. And these are all frogs, in case you're wondering what these are. And these are the older frogs in a dish. So I used to have these dishes full of these frogs everywhere in my lab. They're really fun because you can have many, many of them. And the way that you would do it is that you would inject a female with hormone the night before, and you'd leave her in a bucket near your lab. And then the next morning you would come in and you would massage her, and it would squeeze out all these eggs. And then you could fertilize them, and they were all simultaneously fertilized. So all of these were at the same stage, and they would divide once, and twice, and 3 times until they became a tadpole. So in these tadpoles, I cloned this gene out, which was known to be a receptor really important for blood vessel growth. And when I looked at its expression, and what you're looking at in yellow is the tadpole, but in gray is the expression of the gene. So it is we're literally looking at the RNA. And so this gene is not expressed in the back, in the brain, in the belly. It's only expressed in certain regions. And so I was like, what are all these little dots? And so what the little dots are stem cells for blood vessels, and they're migrating around, and they're trying to figure out the right place to be. So my entire PhD was studying how this receptor, and this is its ligand, or the growth factor that it binds to, how these cells were expressing this receptor and moving around. So these are cross sections through embryos, and this is showing you where the ligand is, the growth factor. It's in this little portion in the middle of the embryo. And these are the progenitor cells that express the receptor. And what I found was that they moved around and they went towards the source of this growth factor. So these cells, this is a little earlier, and they're migrating up here to this source of this growth factor. And then they're forming at this location here what's called the dorsal aorta. And the dorsal aorta is a major blood vessel within the embryo. And really, when I kind of look at my work, this started in my PhD, but I'm still working on this gene now, 25 years later, and I'm still trying to understand how blood vessels go to the right place in the right time. And I'm going to tell you a little bit about how blood vessels actually affected my work in pancreas when I went to my postdoc. But okay, so this was my PhD, and what was life like? You guys probably aren't even thinking, you're just thinking college at this point, but if you ever decide to go to graduate school, the way that it works is that you join a lab, you do rotations, and you end up doing experiments. Your tuition is paid for. So is you have a stipend, so it pays you, wherever you're living in your apartment. So it's really nice, and your entire focus is to do experiments. But you have to do experiments that make sense, right? And so to figure out what experiments make sense, you have lab meetings. And so I actually have 2 of you in my lab. So Kate and Nikita are in my lab, and they come, and we have lab meetings, and we discuss the experiments, what worked, what didn't work. You guys are really in the... What do you think about it? In fact, maybe I'll put you on the spot and just ask you, is this just like so far from what you imagined lab work would be? Nikita?
A little bit.
A little bit? What was different than you expected?
It's a lot more like laid back and discussion based.
Yeah. Yeah, I mean, it's funny. I was a really shy person. I almost didn't go into science because I heard you had to speak in public. And I was just like, I can't do that. But, I decided to be brave and to, do it. So I went to graduate school. But yes, so you have to discuss. You think you're going to go into science and you're just going to be at your bench and nobody's going to bother you. But in fact, you need feedback. So it is more laid back. We talk, we say, what worked, what didn't work. And so this is really, really an important part of the work. So you have to also give presentations, which is what was horrifying me at the very beginning, but you can do it. You work up to these things slowly, and you give feedback. And you guys have witnessed this. All my graduate students and postdocs give presentations, and we look at it and we say, well, what about that? Well, and so lots of feedback. And you want the critical feedback at home before you end up going to a national conference, which is part of our work also. So I go to about, I would say, 5 to 15 conferences a year where I present my work, either in poster format or giving talks, and receive feedback from not my friends, but my competitors who ask me really hard questions. And this may seem all overwhelming, but you build up to this slowly, and I never thought I could do it. But one of the major shifts that is important to know is coming, and I find a lot of graduate students are not ready for this. Right now in your life, mostly what you have to do is learn facts and then regurgitate them, right? I mean, some of your teachers are awesome and they require you to do some critical thinking and have opinions. But mostly it's important that you just learn facts. However, when you go to graduate school, the next step, the rules change completely because we don't know the facts. So nobody's teaching you the facts, you're finding out the facts, and then you got to kind of put them together. So I'm constantly struggling with my graduate students to tell them, I don't know the answer. We are sitting on the edge of knowledge. So you have to figure things out, and you kind of have to put it together, have ideas, and you have to be wrong a lot of the times, but figure out the truth. So it's a real change. I love showing this slide because this is what I show all my graduate students when they join the lab. This is the expected route of your PhD. You choose a lab, you have a project proposed by the PI, a hypothesis is put forth, you do the experiments, you work because you're meticulous and you made A's in high school. You get a result, you expand on the result. There might be a side observation, but mostly you expand, you write a thesis in a big paper, you publish the paper, you graduate, and your parents are so happy. That's what you expect going to grad school. Everybody expects this. But the truth of the matter is much more complicated, because no matter how good you are at working and how meticulous you are, you don't know what the truth is. So you can have wrong assumptions, you can have hypotheses that don't work, and that is okay. That's part of science. And so having the hypothesis and deciding that, oh no, I need to adjust my hypothesis and then do the new tests, this thing can repeat 1,000 times. Lots of different arrows, but point being, in the end, you may write a paper and the entire field may reject it. In fact, many of the Nobel Prize discoveries are not published in the fancy journals like Science, Nature, and Cell. Many of them are published in lower journals and then were only appreciated later. So it's a struggle, but at the same time, it's a necessary struggle, and I think it's always important to point these things out first, and I think it's a lot of fun. What my work does, I have to publish, and so this is one of my very first papers in developmental dynamics in 1997. I was so excited. I'm first author, Ondine Cleaver. That was the guy who taught me, Paul Krieg, and then it went on and on. But papers are our currency. That's the only way you can say I'm good, is I published papers. And my husband's family always makes fun of me for not taking my husband's name. My name is Ondine Cleaver. My husband's name is Thomas Carroll. The schools always get confused that we live at the same address. We are married. But I kept my name because I published 10 papers before I ever married him. And I didn't want to lose that in my publication record. Because really, as a scientist, you end up becoming a number. You are the number of your publications. When I left as a graduate student, I had 10. I have something like close to 70 now. There are some very fancy scientists who have 600 publications. But it is a measure of your ability to think, to put a story together, to tell the story, and then to have it out into a publication. So that's a unit of success in our field. So I graduated, which was great. I wrote my thesis in about a month. I put all my papers together. I put it together. I presented it to my PI, and I graduated, got my degree. It was great. And I thought I knew everything. I was in a small lab with a very supportive PI, and I applied to many different places, mostly around Boston or at Harvard, because my husband, at the time we married, right in our last year of graduate school, we wanted to find a place where we could both do work, and just happens to be Boston has a lot of life sciences, and so we ended up going there. And I applied to a number of labs at Harvard. There was Harvard Medical School and Harvard main campus, and they all said yes because I had 10 papers. One guy in particular I thought was really great, and I'll show you him in a minute, but before I ever showed up, I wrote 6 fellowships for postdoctoral. You have to pay for yourself as a postdoctoral fellow. But anyway, so I wrote 6 postdoctoral, I interviewed with 6 postdoctoral PIs or labs. I wrote 6 postdoctoral fellowships, and I got them all because I had those papers. So it was really great. I ended up being a Damon Runyon Fellow, and this was an amazing environment. But I thought I knew everything, and I show up there, and everybody's brilliant, and they're doing the craziest experiments, and it was so fun, but it was like feeling like you're on top of the world and suddenly realizing that, I know so little. And it was an incredible feeling to be surrounded by people doing amazing work. So the guy whose lab I joined was named Doug Melton. He had been a developmental biologist doing very simple work with frogs, but his son was born with diabetes, type 1. His immune system attacks cells in his pancreas, and so he cannot make insulin. And so his dad said, we're going to cure diabetes. And so his dad, in fact, very famous guy, at the time he was an advisor to the White House when George Bush was there. And his sort of big thing was that he wanted to turn stem cells into pancreatic beta cells. And part of the reason he wanted to do that was because his lab was figuring out that in the normal pancreas, there is no stem cells. You are born with all the cells you're going to have, pretty much. There's a slow rate of replication, but there are no stem cells. So he became a huge advocate for taking human embryonic stem cells. And what that means is going to IVF clinics, and there's frozen embryos that are at that very beginning stage when it's only a few cells, not a baby, few cells, and turning those cells into beta cells. And that's what he wanted to do. So it was really challenging. He changed his lab over 100%. When I was in his lab, his daughter, who was 14 at the time, Emma, he came in one day and said she has late onset diabetes. So both children in his family had diabetes. So we were really amazingly focused. Half of the postdocs that were working in his lab had diabetes. And at first I thought, this is a manageable disease. You don't die from it. What's the big deal? Well, I sat there back to back with a postdoc who had diabetes, who pricked his finger all day long. And you know, he was basically a postdoc. He didn't have very much money. So the little strips, do I have them here? The little strips here, he would cut a single strip into 6 pieces to try and save money on buying these strips. And then he would inject himself in the belly with insulin all through the day. And I realized this is not fun. And if he did not, he gave me a key to his apartment because he said if I don't show up in the morning, I might have had an episode with either too high or too low glucose. He said, make sure send the EMS. And it never happened because he was really meticulous at sort of managing his glucose. But point being, it really sucks. It's a terrible disease. So we want to try and understand how to fix it. And one of the ways that we can do that is by learning about the pancreas. So the pancreas is an organ in your digestive tract. The stomach will turn your food very acidic, but it is the pancreas that will make all the enzymes that will actually digest the food. It's like a tree. It's a fluffy tree with like little tips of the trees are these cells called acinar cells that will make digestive enzymes, which will go down the branches of the tree and be brought into the stomach to digest your food. But about 5% of your pancreas is these little floaty structures called islets, the islets of Langerhans. And these are the cells that are regulating the hormones that regulate the level of glucose. So when you eat a candy or food, your blood glucose goes up. And high blood glucose in your blood vessels is really bad. All right, it tends to damage the fine blood vessels in the tips of your fingers, in your eyes. And so if you're in a let's say a country that doesn't have really good medicine, you can lose fingers, you can have parts amputated, you can become blind from diabetes. In the United States, we're lucky because we have good medical care. But the point is, diabetics don't have these cells, so we'd like to be able to make them. So how do you make these red cells here on the inside of this islet of Langerhans? So when I went to Doug's lab, I showed up and he was out of town. I picked a bench at his desk and he said, when he showed up a couple of days later, he said, well, why don't we talk in a month when you've decided what you're going to do to cure diabetes? And I was like, what? I said, you're a Harvard professor. I remember having this discussion with him. I said, you're the captain of this ship. You know what all the hands in the lab are doing. Tell me what to do. And he says, no. He says, that's not how I do it. He says, I hire the best people. You have unlimited money. He said, you go figure it out. He says, I want your opinion. And I was like, whoa. So my background was a developmental biologist. I like to understand in the embryo how things are coming about. And so this was my first experiment in the lab. And these are cross sections through an early embryo. This is the neural tube, the back of the embryo. The belly is down here. These are the big blood vessels in the middle of the embryo. This is a little earlier, a little later. But the blue color represents where the pancreas is growing. So this was what I decided to do. I said the best way to make a pancreas is to understand how the embryo did it in the first place. And so these are later studies that I did looking at, in green here are beta cells, and in red are blood vessels. And what we ended up finding and publishing in a science paper was that these blood vessels were providing signals to these beta cells that helped them become what they were. And so these are just some of the tissue recombination experiments where what I did was to use cells of the early pre-pancreas in the gut, culture them. They never expressed insulin, but when I recombined them with blood vessels, then they did. The black here is insulin. So what this meant was that the blood vessels were really important for the cell fate of those beta cells. So since then, I've been working on that. This is an early pancreatic blood. The green is the outline of all the cells. It's a complicated looking structure. And understanding how it then will transform into a tree. A couple people have to leave, so sorry, but no problem. How they reorganize is really important because cells talk to each other. They're going to make decisions about what they're going to become. And one of the ways that they make decisions about how they're going to become is arrangement relative to others. Here's an early pancreas in the mouse, and what you're actually looking at is a protein that gets deposited on the inside of the tubes within the pancreas. And so here's a close-up on all of these tubes, and somewhere in here are the cells that are going to make decisions to become beta cells. And they only live in this plexus as the embryo is developing. And then when the embryo is born, this plexus is no longer there. It resolves just into a single tube. So what happens in a special environment? That's one of the things that our lab wants to try and understand. And we'll get into it a little bit more in a minute. So this is what I did as a postdoc. And then finally I arrived and at the end of my postdoc, I had nice publications. I'd been there for 5 years. And I went out on the job market. I applied to 33 places. I got 16 interviews. My husband was doing the same thing. 9 of them co-aligned. And we got offers from UNC, Chapel Hill, Vanderbilt, Cornell, and a bunch of other good places, UC San Francisco. This one was one we didn't apply to. I'd never wanted to move to Dallas. I'd lived in Austin, which is very different style, and I thought Dallas was too stuffy for me. I went anyway, and I just thought, this is amazing. The people were nice, the resources were great, the scientists were happy, and so it ended up being one of the best decisions ever, and now I love Dallas, obviously. But this is what I do. This is my life. Kate, Nikita, you guys can testify. That's me right there, eating my lunch on my lap and on my laptop. I'm in my office all the time because I'm writing grants, I'm communicating with other people, I'm organizing meetings. And the people in the lab, here is Kate and Nikita and the rest of my students, they're the hands, they're doing the experiments. And so us talking is how we decide what to end up doing. And they're learning, so it doesn't always happen right, but that's fine. But they're learning, to ask the questions that we're interested in the lab. I do a little bit of teaching, mostly because I like it, although I don't have to. Most of my job is research and writing papers. So it's been really fun. The best part about the job is that I do exactly what I want to do. I'm asking the questions I want to ask. And so I've told you a little bit about how I'm interested in how cells associate with each other. I want to understand, for instance, how do cells decide to become a blood vessel? This is an early mouse embryo. These are the dorsal aorta. How do these cells decide to attach at these particular locations? How do they know in from out? Oh, 5 minutes left. Okay, but you guys get the idea. These cells are really different from these cells. These are the cells that are going to end up giving rise to beta cells. So how do they do that? How do they pop out? I think the best place to learn, the best place to figure out where that's happening is the embryo, because the embryo does it right. That's why we're all here. We know that cells in the embryo move around. As they're moving, they communicate with each other. This is a face forming in a human embryo. This is a fly forming here. The cells are all dividing. This is a frog, all the cells dividing here. So trying to understand how the DNA is read and interpreted and how proteins are made in each one of these cells is what we do. Again, we would love to be able to make all these tissues since we understand how they, or we are learning to understand how they grow. There's a huge need for new organs. In fact, some of the statistics here, these are the number of people on the transplant list waiting for transplants. Every 10 minutes, another person's added. 20 people die every day waiting for lung transplants, liver transplants, heart transplants. So is it possible to make them? So in the lab, we're doing a number of these techniques. We're doing stem cell differentiation. So that's Annie in the lab, does a lot of this, using organoids to make tissues. We don't do much regeneration, though down the hall people are doing this, trying to understand how to regrow new cells. We can bioengineer new tissues, such as decellularized matrix. And so all of these are incredible methods in the lab that we're learning about. Understanding the 3D anatomy, we also look at kidney and trying to understand how all these cells organize. This is the nephron, the unit that filters the blood. how do you make all those blood vessels in this type of complexity? A certain amount of self-organization occurs, but so I'm just going to skip through these slides just a little bit, but basically say being a scientist is really fun. It's challenging. I present my work at conferences. This is a conference I went to in Greece last fall. This is me presenting students with some of these awards. I'm going to Korea next September. It's really exciting. So I go to about, I would say, 5 to 15 meetings a year. So I love the traveling. And it's not been easy for you women and men. The balance of a career and a demanding, intellectually and time demanding job. This was my number one, Ella Carroll, who's at high school here. This is my number two, Cormac Emmett. I had them while I was a postdoc and while I was a new faculty member. But the answer is it can be done. What I learned is really to think long-term over short-term. Any job is hard. You think Katy Perry goes up and just starts singing? No. She has to practice a lot and sweat and compete. You think any of these financial analysts who make millions of dollars, it's easy? No, it's hard. So anyway, work hard, be creative, be adaptable, be tough. Nancy Pelosi said, wake up, eat your bowl full of nails, and put on armor and go to work. And that's what I do at the lab. And I have a great time. Approach everything with a sense of humor. Do what you love, I think is most important. And get out there and do great things.